In the last couple of chapters, we've talked about the process of gene expression, so going from our genetic material to our phenotype and how that happens. We talked about the process of cell division and a little bit about reproduction. So now we're going to continue this idea and talk about how specific chromosomes and specific genes are passed on and how those genes are then represented in offspring. So you've probably noticed that you and your family members resemble one another. You probably look like a bit like one or both of your parents. Maybe you look a whole bunch like one and not much like the other. But the reason for this is that both of your parents, your biological mother and father, contribute to your genetic makeup. Now, sometimes this is good, sometimes this is bad. So as an example of something bad, let's talk about fish odor syndrome. So this is something I never heard of until I read this book. Basically, the poor individuals that inherit this disorder smell like rotting fish. They are supposed to have a gene for an enzyme that breaks down a chemical in our body that smells like rotting fish. So we all have this chemical, but most of us have the functional gene that produces a protein that breaks this chemical down. These individuals that inherit this disorder from their parents don't have that, and so they end up smelling like rotten fish, and there's nothing they can do about it. Their breath smells like fish, their sweat, their urine, and they can't wash it off because this is basically a byproduct of their normal bodily function. So you can imagine that this is a problem for individuals. Now, most of our genes are not a problem, especially when they're functioning correctly. And the cool thing about genes is they all follow a particular type of pattern when we look at inheritance. And this goes back to the fact that we have 23 chromosomes, and these chromosomes come in pairs. So one of each pair comes from mom and one of each pair comes from dad. And then we have a total of 46 individual chromosomes, 2 copies of each type. Remember that one of those pairs is our sex chromosomes. So we have 22 autosomes and then 22 autosomes and then a sex chromosome from mom, 22 autosomes and then a sex chromosome from dad. So keep that in mind as we're talking about this. For all of this discussion, we are going to be talking about the individual copies that one receives from mom and one receives from dad. So up here we've got mom and her chromosome pair, dad and his chromosome pair. They're going to be separated in meiosis, and then when an egg is fertilized by a sperm, we get our diploid set of chromosomes back. So remember that each individual is going to have 23 sets of these guys. So remember that all of our cells except our gametes are diploid. This means 2 copies of each type of cell. So our diploid number for humans is 46. When we produce our eggs and sperm, we become haploid. Our haploid number is 23. Remember that the location of a particular gene on a chromosome is called a locus. And for any gene, we each carry 2 alleles. Now we can have 2 of the same alleles, or we can have 2 different alleles. Remember, alleles are versions of the gene. So this chapter really focuses on heredity or inheritance, and how offspring actually inherit genes from their parents. So the study of heredity is simply the study of the passing of characteristics from parents to offspring. So we've got a couple examples here. Again, individuals often look like one or both of their parents. You can see specific features or characteristics in every individual that you can trace to their parents and to their siblings. So this diagram here is just a sort of a summary of the fish odor syndrome. I'm not entirely sure why it's on this page. That was a bit of a screw up apparently. But anyway, we see how an individual might actually acquire 2 defective copies of this fish odor syndrome allele. So over here we've got mom. She's got a normal version and a defective version. Dad's got a normal version and a defective version. So remember, these split up in meiosis. Let's assume that this poor individual gets mom's defective version and dad's defective version, and then during fertilization those gametes come together to be diploid again, and now this poor individual has 2 copies of the defective gene, which means that they have this fish odor syndrome. However, neither of the parents had this syndrome, and that's because they only had 1 bad copy. So we'll talk about why sometimes you need 2 bad copies for something to show. Sometimes you only need 1 good copy to cover it up. You can sort of think of this as genes having different weights or different effects on individuals. So one way that we can see heredity in action is by looking at selective breeding. So humans aren't very good at selective breeding. We are generally not picking individuals so that our children have specific characteristics, although maybe we are just a little bit unconsciously. But you can really see this selective breeding when you look at things like dog breeds and things like horses, cattle, any domesticated animal. You're going to see evidence of selective breeding, which is where people are breeding those animals for specific traits that they want. Now, you can also do this with plants. This has been done with agriculture for many, many years, and even just selecting for things like flower color in your garden. You can do that too. You just basically follow these traits through generations and select for the individuals that have the most desirable trait and breed them again. So once breeders recognized this ability to select for heredity, they started selecting individuals with desired traits to breed with one another, and therefore sort of reinforcing the traits that we liked. Examples of this are sweeter corn, loyal dogs, docile livestock, beautiful flowers, things like that. The thing is, though, is people didn't understand how this worked for a really long time. They were practicing this and doing this, but they didn't really understand the mechanism behind this heredity. Now, we are at a time when we're on the other end of this. We understand the mechanism. You guys understand the mechanism. We talked about gene expression. We've talked about cell division. We've talked about sexual reproduction. And so you guys understand how this works. But even less than 150 years ago, people didn't understand how this worked, and they didn't understand the molecular basis for this heredity that they were seeing. So let's begin our discussion of heredity by looking at single gene traits. So these are characteristics that are controlled by 1 gene. Now remember, each individual has 2 copies of this gene, and sometimes they're the same, sometimes they're different. But even if they're different versions, they're still coding for the same trait. So for example, we've got long hair and short hair. These traits are, fur length is controlled by 1 specific gene at 1 loci or locus, and then depending on the allele that they have, they're either long-haired or short-haired. Same thing with white-haired cat or colored-haired cat. This is controlled by 1 gene. A single gene controls fur color, and then you have multiple alleles that give you different fur colors. Now we look at single gene traits because they're easier to study. And it's easier to follow the pattern of inheritance when you're only looking at 1 trait. I want you to keep in mind, though, that most human characteristics, and even most characteristics in other animals, are actually influenced by the interaction of multiple genes and even the environment. So even though we're studying some really cool examples of single gene traits, and we'll look at lots of different ones, it's often not as simple as that. So once we get through these single gene traits, we'll look at some other instances of gene interaction, environmental effects, and things like that. So one of the most important people in the history of genetics is Gregor Mendel. So Gregor Mendel was a monk in the Czech Republic living in the mid 1800s. And he studied very extensively heredity in pea plants. And he did this because he wanted to ask the question, what do parents give their offspring that confers similarity? So why do offspring so closely resemble their parents? And so before him, the prevailing idea was that inside the head of the sperm was a little tiny human. I find this image really disturbing. I've seen some other pictures that are even worse. But basically, they thought that a little tiny human was in the head of the sperm, and when somehow it eventually grew into a full-fledged baby and then ultimately a human. But Gregor Mendel wanted to look at this question a little more closely and just sort of see if he could figure out why offspring resembled their parents. Now, if you think about it, if there's a little tiny human inside the head of a sperm, that little tiny human has to look exactly like dad. But when you look at individuals, they look like both parents. And so Mendel sort of realized that you get information from both parents. Now, the reason that his work was so important was that he really did the scientific process well. He had these really great controlled experiments. He took really great notes. He had a lot of data. And because he did such a good job at designing his experiments and collecting his data, he was able to really see some patterns in inheritance and really elucidate for coming generations what was actually happening. Now, one thing that's important to remember here is that when individuals when our offspring are produced, there's something called blending that does not happen. So blending is the idea that you get a little bit of mom and you get a little bit of dad and your characteristics are sort of the in-between. Now, the problem with that is that most traits are not in between. And so we don't actually see this idea of blending. What we actually see is specific traits from mom and specific traits from dad passed on to offspring. Now, you guys know from the last chapter that you can get new combinations and things like that, and that's why individuals look a little bit different. But what you don't see is sort of intermediate traits between parents because those genes have sort of blended together. So there were three features of Mendel's fantastic research that were really critical to his success and his understanding and being able to hypothesize the basis for heredity. The first is that he chose a good organism to study. Now, today, this would probably be called a model organism. A model organism is just an organism that's kind of a good representation of other similar organisms. They're usually small, they're easy to take care of, they produce a lot of offspring, they have short generation time, so you can get a lot of information from them very quickly. The fruit fly is a really good example. The lab mouse is another one. There's a really cool little fish called a danio that is see-through. You can see all of its organs. And so there are different reasons that you would use these organisms to study different things, and there are lots of different ones. But the early one in genetics was the pea plant. The pea plants produce lots of seeds. They're really easy to breed. They're generally small plants, and so you can have a lot of them in a small area. The next important feature of Mendel's research was that he chose easily categorized traits. And I'll give you some other examples in a little bit, but he chose things that were very easy to see. So one of the big ones that he studied and that we talk about a lot are our flower color in these pea plants. So purple flowers and white flowers. And he chose plants that started out with only those features in order to focus on them. And that made it really easy. He also chose characteristics that only had a couple of versions. So we'll see a little bit later that sometimes you can have many, many alleles, different types of colors and things like that, but he chose traits that were easily distinguishable and didn't have a lot of different categories or different types of traits. He was also able to sort of isolate his populations and therefore have distinct populations that were not interbreeding. And part of this was due to the ability to make sure that the lines were sort of pure. I use that with air quotes, so that he only had particular traits in particular family lines, and therefore there should be no additional alleles within these particular groups. Now, this is a concept called true breeding. It means that he started with plants that always produce the same offspring. And he sort of knew then that no matter what this parent, no matter how this parent was passing information on, it was always going to pass on that trait. So for example, over here we've got purple flowers. You cross purple flowers for no matter how many generations, they always produce true breeding purple flowered plants. Same thing over here. We've got white flowers. No matter how many times you cross them, you end up with white flowers. Now, this was really important because it allowed him to know that those plants could only pass on that genetic information. The only thing they should be able to do is pass on a purple or a white allele. So it's important to keep in mind that we are sort of at the time where hindsight is 2020, and we can look back and be like, oh, duh, that makes so much sense. We have this understanding of chromosomes and sexual reproduction and sperm and eggs. But keep in mind that Mendel was doing this before there was no knowledge or understanding of chromosomes. So all he could really say is that parents are passing on something, more air quotes. He didn't know what it was. He just knew that somehow this trait was being passed on, and always in a particular manner. So all of his work amounted to crossing and breeding thousands of plants over the course of his studies. So many years doing this. And that was really important because it's kind of all about the numbers. The more data you have to support your hypothesis, the better off you are. And if you continue to support that hypothesis, then it lends better credibility to your experiment and to your hypothesis. So in breeding all of these plants, looking at these very distinct characteristics, starting with true breeding plants, he was able to describe the laws of inheritance that have remained laws today. So basically, you could really say that that Mendel was able to describe the theory of inheritance, and these patterns have been continuously supported ever since then. Now, that doesn't mean that we haven't discovered new variations of this, new ways that traits interact, but at the time, this was really groundbreaking. So let's talk about Mendel's particular experiments. So we've got this image here. This is not in your book. This is in a different book that I have taught with. But it kind of shows how he was able to do controlled breeding. Now, we can't do this on people. Controlled breeding programs on people are frowned upon. And so that's why using something like a pea plant or a lab mouse, things like that, is much more desirable. People don't like it when you tell them who they need to breed with and how many children they need to breed. So being able to cross plants really easily, get a large number of individuals from any cross is really important. So basically, he could take off the male or female parts of the flower. The carpel here is basically the structure that holds the egg. The stamens hold the pollen, and so he could brush pollen from one plant onto the stamen of the other. And so he knew which traits he could be looking at, which ones he could study. And so here we have the purple flower crossed with the white flower. We produce the peas in the pod. Those are the offspring. that are going to grow into the plant. So in this case, we have 5 peas, so we have 5 individuals from this particular cross. Now remember, not Morgan, I'm sorry, Mendel was looking at specific characters in each of his experiments. So a character would be equivalent to a gene today. So the gene for flower color, the character of flower color. And then he was looking at different variants or traits, trait being synonymous with our current word, which is allele. So sometimes I'll say character, sometimes I'll say gene. I try not to, but if I do say character or gene, they're kind of the same thing. I probably won't say trait very often. I'll probably switch over to allele at this point. So remember, one of the important things in Mendel's work was that he was using true breeding plants. So these are plants that no matter how many times are crossed with one another, they only produce the particular trait that they possess. So purple true breeders always produce purple flowers. So he performed these fantastic hybridization experiments. Hybridization is just when you cross 2 true breeding varieties. Now this is important because he knew that he was starting with populations that only produce purple or only produce white flowers. So those starting populations are called the P generation or the parental generation. Not rocket science here. This is just botany. Parents, offspring. P generation is your parents. After that, things get a little more complicated, and we call the first generation of that cross the F1 generation. These are your hybrids, the result of crossing those true breeding parents. F here stands for filial, or meaning brother or sibling. So this is the first sibling generation. Now keep in mind that he would have gotten lots of peas from this cross, so lots of flowers. There's only one shown for simplicity, but this guy would have had lots of brothers and sisters, or whatever you call a plant that has both carpels and stamens. I don't know. But after that, he would cross those guys, either cross 2 individuals from that generation or self-pollinate those individuals and produce the F2 generation, or the second filial generation. Now it's important to note here that in the F1 generation, all he got was purple flowers. So I said that the only thing the white flower could pass on was that white allele, but then that white allele disappeared in that generation. Now that's important for sort of understanding what's going on. Down here in the F2 generation, we see there's still purple plants, but we see we've gotten back that white flower. Oops. All right. So there were a couple of important observations for starting with these true breeding parents. The first one was that the F1 generation lost one trait. Now this is important for 2 reasons. One, because it means there's no blending. So if these traits were being blended rather than distinctively inherited, we would probably have sort of a light purple here, somewhere halfway between purple and white. Now the second important thing that happened is that that characteristic that was lost actually reappeared. And so it turns out that it really wasn't lost. It was simply masked by the other trait. And it also suggested that this individual, even though you can't see it, actually had the allele for the white flowers, that the purple was covering it up. And then in the F2 generation, some individuals were able to sort of reacquire that white flower allele. Another important little bit of information here is that Mendel was using thousands of plants. We can see here we've got almost 1,000 plants just from this purple and white flower cross. Those huge sample sizes are really important for the validity of this experiment. If you do this one time and you happen to get all purple flowers, I mean, that probably doesn't really tell you something. But when you do this with hundreds of crosses and hundreds of hybridizations and you continuously find a large number of individuals exhibiting this behavior, it gives credibility to your experiment. Now, the other thing that's important to note here is that in that F2 generation, the traits were almost always, or not almost, were always displayed in roughly a 3-to-1 ratio. That means for every 3 purple flowers, you got 1 white flower. So Mendel observed 7 total characteristics, and he noticed that all of them followed this roughly 3-to-1 ratio. Now that'll be important a little bit later when we get into sort of the probabilities of inheriting different types of information. But I want to sort of just show you how many plants he was actually looking at. I mean, 6,000 plants that exhibited a yellow seed color, 2,000 exhibited a green. And all of this, he ended up with roughly a 3-to-1 ratio. Now, the reason this isn't exactly 3-to-1 every time is that genetics is subject to chance. So think of this as a coin toss. You can flip a coin 10 times and you would expect to see 5 heads and 5 tails, but sometimes you don't. Sometimes you see 4 heads and 6 tails. Let's say you flip it 100 times. You would still expect 50 and 50, but you might get 48 and 52. It just depends on the day and the flip and all of that. So genetics is basically a study in statistics. We won't go into a lot of statistics here, and statistics and probability, but we will use the ideas a little bit. So just keep in mind that you might say that you expect 3 of your offspring to be purple, but you might not actually get that. So just keep that in mind as we continue talking about these different inheritance patterns. So let's sort of summarize what Mendel was doing here and what he observed. First of all, he observed that the heritable factor that controlled the white trait wasn't lost. Instead, it was sort of masked by the purple trait. It was covered up by the purple trait. So Mendel gave this idea a couple of names. He called the trait that covers the other one the dominant trait. And he called the one that was masked or hidden the recessive trait. Now we'll see in a few minutes that this is related to the idea of being homozygous or heterozygous. So you may remember that from earlier discussion. So keep in mind that this will come back to that. Something else that's important to note here is he noticed that some of these purple flowers were no longer true breeding. So they would, if he bred these purple flowers to one another, sort of a next generation, you would occasionally get some white flowers back. So that suggests that these guys are hiding that white trait as well. So this guy's hiding the white trait. This guy's hiding the white trait. These guys, however, if you bred these guys to one another, they continued to be true breeding. They no longer had that purple allele. All right, so just a real quick review of some vocabulary. Remember that a gene is a DNA sequence coding for a specific protein, and that alleles are alternative versions of those genes. So alleles are going to account for a variation among individuals. And keep in mind that each individual receives 2 alleles, one from mom, one from dad. Now these 2 alleles could be the same allele, they could be different alleles. So there were 3 important ideas that came out of Mendel's research. Now, keeping in mind that Mendel didn't know what was being passed on, but because we do, we are going to use our terms now. So 3 important things that came from Mendel's research. One is that each parent puts out a single set of genes into the sperm or the egg that it makes. Upon fertilization, offspring receive 2 copies of the instructions for any trait. And then finally, that observed trait that an individual possesses depends on which 2 copies or alleles of the gene it inherits from the parents. Now, an individual can receive 2 of the same copies, the same type of copy of an allele. That's a condition called homozygous. Remember, homo meaning same, zygous meaning zygote meaning fertilized egg. That's just a fancy term for fertilization. But an individual can receive 2 copies of the same allele, or they can receive 2 different copies or 2 different versions of the same of a particular allele. Now, we saw this when we were looking at sickle cell in our lab. Individuals could be homozygous normal, homozygous for sickle cell, or they could be heterozygous, where they produce both type of protein. So one last thing that was important from Mendels research was the idea of what we now call the law of segregation. Now, the law of segregation says that you've got 2 copies of each allele, but those copies are separated, that's what segregation means, into each their own cell during meiosis. So we saw this during meiosis. It happens when homologous chromosomes separate. So up here we've got an individual who is heterozygous for flower color. So they've got 1 purple and 1 white allele. During meiosis, those alleles are separated or segregated. So each gamete either gets a purple or a white. Same thing happens in the other parent over here. Each gamete gets either a white or a purple. So the law of segregation says that each sperm or egg is only getting 1 copy, and it can be either copy. So then when we fertilize that egg, we get back our diploid situation, and we can see now the combinations that we can get. We can get homozygous white or homozygous recessive if the 2 white alleles come together in fertilization. We can get homozygous for purple or homozygous dominant if the 2 chromosomes with purple alleles come together, or we can get 2 different situations of heterozygous individuals. So in this case, it looks like let's say this side was mom, the white allele comes from mom, purple from dad. The other way we can get heterozygous is purple from mom and white from dad. So another way to think about this is that if a parent is heterozygous, half of their gametes are going to get 1 allele and half of their gametes are going to get the other allele. So there's sort of your flip of a coin. If you're heterozygous, there's an equal chance of either of your 2, sorry, my dog is sneezing, either one of your chromosomes to be separated into a particular sperm or egg. Now, if you're homozygous, these individuals down here, the only option is for them to pass on the 1 allele, sorry, dog's being wild, 1 allele to your egg or your sperm, so to the next generation. So this individual can only pass on the white allele. This individual can only pass on the purple. These 2 individuals, when they produce sperm or egg, could pass on either allele to the next generation.